Right. I will explain repeatedly. Okay. Right. So first, amino acids are inactive state in cytoplasm. They are activated after react with ATP energy. Amino acid transferase enzyme. Amino acid synthase enzyme. In the presence of amino acid synthase enzyme, now inactivated amino acid activated. Right? Is it clear? First step clear?
Yes, sir. Yes, sir.
Right. Now, so and after amino acid activated should be activated. The amino acid attached, which one carries the amino acids? Which RNA type carry amino acids?
mRNA, TR, yes, sir. In this transfer.
T means transfer RNA. Then transfer
Yes, sir.
RNA
Yes.
activated.
Hmm.
Transfer RNA activation of the acid to the tRNA CCA end. Do you know tRNA structure?
Yes, sir.
Structure,
Slowly model.
right? This is CCA end. At the C, this activated amino acid attachment. Now the amino acid tRNA complex form. Now automatically this tRNA activated with the active amino acid. Right? So this is the first.
Unable to get you, sir. Your voice is breaking a lot.
Right? No problem. No problem. And again. Right? So activation. Right. Please be silent. Please be silent. For the activation of amino acids, mainly first with help of ATP and amino acid synthase enzyme, the inactivated cytoplasm amino acid, the total different 20 types should be activated. And this activated amino acid attached to the tRNA CCN. Now the tRNA amino acid complex formed. Now tRNA activated. So the first step complete. Now the initiation begins. Again divided into initiation step. Again divided into mainly first 43 years complex formation. 43 years complex formation and second one attachment of mRNA with 43 years complex. Third, scanning. Fourth, large subunit attachment. Large subunit attachment. Right? So, first of all, the amino acid, first step clear? Hello?
Yes, sir.
So first, right. Okay, no problem. If my voice not clear, I will explain again and again also. You don't worry. Right?
Okay.
So first of all, is it clear? First step clear?
Yes, sir.
Yes, sir.
Right. Now the second
Initiation is divided into 4 steps.
initiation. Initiation translation process. For initiation, the mainly requires initiation. For the initiation, again several substeps. First of all, formation of 43S complex. Formation of 43S pre-initiation complex into better. Pre-initiation complex. So that means 43S pre-initiation complex and means with 40S subunits. Right. Next step. These prepared pre-initiation 43S complex attached to mRNA mRNA. Next, third one, scanning. Fourth one, large subunit attachment. Large subunit attachment. Now let's see the first one, 43S pre-initiation complex. Before that, first 40S subunits. Initially, ribosome in eukaryote, what type of ribosome existed in cytoplasm? Hello?
ATS.
In ATS subunit. The initial stage, this ATS subunit should be disassociated, should be separated, right? Then only the 40S and 60S separated and attached to the mRNA strand. Isn't it?
Yes, sir.
Hello?
Yes, sir. Yes, sir.
Right. So first, this ATS subunit disassociation, ATS initially disassociation into 40S and 60S.
60 years of unit.
And this is carried out by initiation factor F6. Initiation factor F6. With help of initiation factor F6, the ribosomal ATS subunits, ATS ribosome separated into, disassociated into 40S small subunit and 60S large subunit.
That's a unit.
Right? Next step. This 40S subunit, small 40S subunit binding with initiation factor, eukaryotic initiation factor 3 and eukaryotic, write down this all. Please write down. Right. 40S. First attach with initiation factor 3 and initiation factor 1A. Initiation factor 1A and initiation factor 3. Right? Next. These 40S subunits with initiation factor 3, initiation factor F1A. Now, next one, just wait. Ternary complex formed. Next, this attach with ternary complex. The ternary complex, nothing but simple. The ternary complex formation already activated tRNA present, right?
Activated tRNA with amino acids already in first step it is ready. And this activated tRNA with help of initiation factor 2, eukaryotic initiation factor 2 plus activated tRNA with GTP energy, this total structure called as ternary complex. Now this ternary complex prepared, and before that 40 years initiation complex prepared. Now this both together form as a 43 years pre-initiation complex. Is it clear?
Yes, sir.
Right, simple.
They form the 43 years
Let me
pre-initiation.
Right, pre-initiation. Let me explain again.
Okay.
Right. First 8 years, first clearly, 8 years ribosome dissociated with help of initiation factor 6. Dissociated into 40 years and 60 years. The first of 40 years, now for example, this is 40 years subunit. In 40 years subunits, there are these sites, first elongation site, sorry, exit site, acyl site. The first, the exit site, E site, this is E site, exit site attached with IF3, this one, elongation factor F3, this region, and EIF1 initiation factor 1A attached to the acyl place, right?
Yes, sir.
Before that, remember in ribosome, three regions are clearly present. The three regions orderly, for example, this is ribosome 1, 2, 3. First, exit site, E site, P site, peptide site, amino acyl site, these three sites. Now in this, already the E site and A site attached with initiation factors 3 and initiation factor 1A.
Oh.
Right? Only which place is empty?
P side.
P site is empty. Right. P site is empty. Right? Now only the remaining P site attached with ternary complex. Already told you how the ternary complex formed. Ternary complex formed. Tell me how the ternary complex formation. Ternary complex. tRNA. Right, tRNA, first amino acid tRNA, already told you the activated, how the formation of activated amino acid tRNA complex, right? tRNA, right? Amino acid, for example, 1. Generally methionine. Generally, first amino acid is methionine. First amino acid in eukaryote, methionine. So that was the symbol M. And this is brings towards this complex, that is 43 years, that means the 40 years complex, the 40 years subunit complex reached this tRNA with help of how this tRNA, activated tRNA, reached to the P site.
Yes.
Which initiation factor involved?
Tertiary complex bind.
So actually in tertiary complex means tRNA, this tRNA attached with initiation
GTP and CAF2.
initiation factor 2 with help of GTP energy. These two are involved. To transfer the tRNA. Please be silent. To transfer the right to P site. Okay. Now
Okay,
this
sir.
ternary complex, this total region called as ternary complex. Now this ternary complex reached towards the 43S subunit. Sorry, 40S
Yes.
subunit, 40S
40
subunit.
years.
Now automatically it becomes 43S pre-initiation complex, right? Now 43S pre-initiation complex ready. And what are the other requirement? Okay, one subunit present and one subunit present, all some initiation factors also present. For prepare the amino acids, we require the mRNA strand. Where the mRNA strand? Now this entire complex, entire complex attached to the mRNA strand. Attached to the mRNA strand. Now, what are the involved? To mRNA strand attachment with help of the 43S pre-initiation complex. They are mainly here involved initiation factor F. Can I rough? Have you written all?
Yes, sir.
Can I rough?
Yes, sir.
If you follow the book, there is
Okay, sir.
okay, right. In initiation factors, the first step clear or not?
Yes, sir,
43
clear.
years initiation complex formation,
Clear,
right?
sir.
Right.
Yes, sir.
Next one. A simple activation of mRNA. Activation of mRNA or mRNA strand attached to the 43S complex. Right? Attachment of mRNA with 43S complex. Right? Then only the mRNA strand activate. Now this is carried out by initiation factor, eukaryotic initiation factor 4, initiation factor 4 F4, initiation factor F4 A, F4G, F4E and F4F, they are all involved. They are involved, right? First, let me explain one by one. The first 4F factor, which is composed of E factor and F factor, they are all. First, F1, this F factor, cap complex, involved in cap binding complex. Cap binding. Do you know the mRNA strand cap region? Right? First, mRNA cap region recognized by initiation factor F4E. Sorry, initiation factor F4F, which binding the cap binding. Now, the mRNA attached to this region. Next, initiation factor F4E. Initiation factor F4E. Recognition of cap. Recognition of cap. And apart from the other initiation factor F4B also present. B also present. The initiation factor B recognition the starting codon AUG. So they are the process. The first of all, the initiation factor F4 involved in activation of mRNA. For this first, recognition cap region with help of initiation factor E and this binding this initiation factor binding with help of initiation factor F, initiation factor F4B, recognition starting codon AUG. Recognition starting codon AUG. Actually, this recognition AUG portion is considered as scanning. Is considered as scanning. That is the next step. Considered as scanning. In scanning, first of all, first step in scanning, the sequence of upstream of AUG is complementary to ATS rRNA, which act as a consensus sequence for recognition of AUG codon. AUG codon, it is known as Kozak sequence, right?
White color tibbi bond.
Hello? Any doubt?
No, sir.
Let me explain again. First of all, the first two step clear. 43S initiation complex formation, and second
Yes, sir.
activation of mRNA in the single word. Activation of mRNA by which factor? Activation of mRNA F4, initiation factor 4, with subcomplex subfactors are A, G, E, F also present, right? So anyway, after mRNA activation, so now the 43S complex present and activated mRNA present. For initiation, one of the initiation codon present. So to recognize the initiation codon, first scan the mRNA strand.
Yes.
The sequence of upstream of, first of all, before finding AUG, some complementary sequence recognized. This complementary sequence existed upstream of AUG. What is upstream and downstream? For example,
Left side.
for example, here this is the mRNA strand.
Yes.
This is starting point. Where the upstream and downstream? This is left.
Left side.
This is right. Where the upstream and downstream? Left or right?
Left side. Left.
Left side upstream. Remember this one upstream. Right side downstream. Downstream. So from the left side, upstream side, consensus sequence. Consensus sequence means almost repeated sequence. Tata box, TA, CC, TA, the repeated sequence. Thus repeated, the specialized sequence, that is specially packed sequence. The specially packed sequence called as Kozak sequence. Called as Kozak. Kozak sequence, right? Kozak sequence. Now, with help of Kozak sequence, the AUG sequence identified. Already told you the AUG sequence identified with help of initiation factor F4B. F4B, right? Now, initiation factor also ready. Sorry, AUG initiation site. Initiation site also ready with in the form of AUG. What is the next stage? What is the next stage?
Scanning.
Scanning.
After scanning, the AUG recognized. The scanning means you should be scanned where the consensus sequence are present on the mRNA strand. First of all, actually, just to bind it. Just to bind that cap site only. First recognize the cap, right? The recognition cap, after recognition, binding with F3S complex. After that, scan the mRNA where the consensus initiated sequence are present. After scanning the consensus sequence, that is called Kozak, because this site discovered by such scientist named Kozak. That's why the name given as a Kozak sequence. So that is identified the AUG with help of initiation factor F4B. Now the exactly settled at AUG place. The F3S complex settled at AUG place. Right? Is it clear?
Yes, sir.
Right. Next one, 40S okay, mRNA okay, tRNA okay, and remaining only one remains, that is large subunit, attachment of large subunit, right? Okay, is it clear?
Yes, sir.
Yes, sir.
All right, let's see how the attachment of large subunit. Last step in initiation, attachment of large subunit. Large subunit. Let me draw here the simple diagram. Please draw this. For example, somewhere there the capping or here AUG codon. (43S). Already told you the AUG. Here methyl group. Methyl amino acid. Right? This is acyl. Already told you this is amino acyl site. This is exit site. P site. This one is P site. After scanning, P site and AUG settled at a time. Right? Is it okay? Now attach to the large subunit, right? Now attachment of large subunit. Now large subunit is nothing but the 60S subunit to the 40S complex with help of initiation factor F5 or initiation factor 5. Initiation factor. This is, for example, somewhere this initiation 60S with help of E initiation factor F5 raised to the 43S complex. With help of initiation factor F5 reached to the 40S complex. Now it becomes complete 8S complex. 8S initiation complex. Right? So after that, right? So large subunit attached to the mRNA strand. Right? Hello?
Yes, sir.
Right. First of all, let me clear one by one, stepwise. In first step, in initiation of translation, initiation step, what would happen? First step.
43 is
Right,
initiation.
43S initiation complex. In 43S initiation complex, also again two stages in orderly. First one, 40 activated 40S complex. This is 40S. This 40S please be silent. Hello, please don't make noise. This 40S subunits activated with help of initiation factor 3, initiation factor 1A. Right. So these two mainly attached to the site and the site. Right. After in second, ternary complex formed. The ternary complex means the tRNA. This activated tRNA with help of initiation factor 2 and GTP energy. This activated tRNA reached the AUG site. Reached the actually not exactly AUG site, but P site, but not actually P site, but not recognized exactly. Not recognized exactly, but it is clearly exactly recognized after scanning. Of course, it is arranged at P site, but not find out the AUG initially. After scanning, the AUG recognized with help of Kojak sequence. Then only the P site AUG, these both are settled together. Right? So next, mRNA strand attached to the this 43S complex. How this attached? How the mRNA activated? What are the factors involved in? Hello?
Yes, sir.
What are the factors involved in activation of mRNA strand? Are these
F4
attached?
F4 factor with subfactors.
F4 factor.
What factor? What factors involved?
F4 factor involves with subfactor A, G, E, and S.
Right. Initiation factor F4. Initiation factor 4. First of initiation factor 4.
Yes, and
The
subfactors like
subfactor G, E, F also present.
A, G, E, F. Yes.
Right. After that, finally, the large subunit attached to the 40 this mRNA strand. This attachment with help of
Yes.
initiation factor 5. Initiation
5.
factor 5. Right. Now, so this P site with tRNA, methionine amino acid tRNA, A site empty and E site empty, right? Is it okay? But remember,
Yes, sir.
the entire translation process, the very complex method is initiation. Very complicated. Be careful. Very complicated. If you not understand, please ask me and I will again explain. Is it clear first?
Yes, sir.
Only if you understand initiation, the further steps would be easy, such as elongation and termination. Without concept, without understanding of initiation, the rest of the two, nothing. You can't understanding the rest of the two. Okay, all of you.
Okay, sir.
All of you clear, right? Right. Next step. So this is the first step is activation amino acid. That the first one that is also completed. Second, initiation also completed and initiation substages also completed. Initiation
Yes, sir.
substages in order they also completed, and the third stage is
Elongation.
right, elongation. Now the elongation, the simple elongation process. Just wait.
attachment of amino acid with tRNA to A site.
Yes.
This is carried out by initiation factor 1 and involved ATP energy. Involved ATP energy. Quite simple. So this is one ternary complex. The same way another ternary complex also formed with another tRNA, another amino acid. For example, this is M-methionine amino acid. And second is serine, for example, serine amino acid, something. We don't know, some for example. So this is the second amino acid. Second amino acid. This is M1 first, and this is second amino acid. The second amino acids also with attached with tRNA same CC end. Same CC end. Anticodon region. Anticodon region means, for example, here AUG. The AUG anticodon here, what should be? AUG, opposite A. Hello.
Use of
Uracil.
adenine.
Adenine.
Next adenine. Next cytosine.
Cytosine. Now UEC anticodon recognize only methionine, not others. Only methionine. Like this, here also another codon there. For example, GCC. What should be the anticodon? This side should be. This is mRNA sequence. GCC is the mRNA next sequence. Now, what should be the anticodon sequence?
CGG.
CG. Now, this CG code translate the only amino acid. That is amino acid. Second amino acid. Now, the same way, the second amino acid attached. This second amino acid with tRNA that brings towards the which side empty A side.
A
A
site.
Amino acid site, right? This for transport of the second tRNA with amino acid to the A site, here involved initiation factor 1. Elongation factor 1 and ATP energy. Elongation factor 1 and ATP energy. Now reach the amino acid site. Now P site, A site, both are filled with tRNA amino acid. Which site is empty? Which site empty in both? How many sites in ribosome? First of all, let me clear.
3 sites.
3 sites. E site, P site, A site. The first of all, P site filled with tRNA. That means, please be silent. Please be silent. Hello. That means you should remember the first amino acid should attach at P site only. First one P site.
Yes.
Already P site existed. The next coming tRNA first reach A site and translocate to P site.
P site.
This would be continued. Okay.
Yes, sir.
So now what I'm saying, E site empty, P site with first amino acid, methionine, and A site second amino acid. Someone amino acid. Now next is peptide bond formation with help of peptide transferase enzyme. To formation peptide bond, no energy required. This peptide transferase enzyme existed in 28S rRNA subunit. That means actually in 60S
in 60th subunits also already given page number 81. All of you open, turn on page number 81. Turn on page number 81. Hello?
Yes, sir.
Right. In page number 81, clearly given the diagram. Right. This is 80S ribosome, 60S subunit, 40S subunit. Again, this 50S subunit made up of 28S rRNA, 5.S rRNA, 5S rRNA. Right. These 3 units. Again, these 3 rRNA and almost proteins, 49 proteins. 49 proteins. That means the each and every subunits made up of RNA and protein. Here also small subunit, 18S ribosome, only 18S ribosome and almost 33 proteins. 33 proteins. And peptide transferase enzyme existed only in this region. Existed 28S region only. Right? Next. 28S region only. Right? So the second step also clear in elongation. First of all, elongation, attachment of second tRNA to A site, the first step. Second step, bond formation. Third step, translocation of ribosome. For translocation, the required energy and elongation factor 2 also required. And finally, released of tRNA. Simple. Right. Second step, elongation. Elongation also stepwise. First, amino acid 2 plus tRNA attach A site.
Yes, sir.
Second, bond formation, peptide bond formation. Right. Third one, translocation. Release. So first completed. Site attachment 2. And second also complete. I will tell you how the bond formation formed between first amino acid to the second amino acid. So anyway, remember here the one peptide bond formation between first amino acids to second amino acid. Next, translocation. Translocation of ribosome required energy. Here bond formation, no energy required, no factor required. But translocation of ribosome, translocation means moving of ribosome, the energy required, ATP is required, and also elongation factor 2 also required. Elongation factor also 2 required. Now finally, release of tRNA. Release of tRNA means, first of all, these 4 steps clear. Hello?
Yes, sir.
Right. Right. Now, shortcut tRNA with the methyl group. This A site, same tRNA with the second amino acid. Here, first amino acid, second amino acid.
Your voice is breaking, sir. Unable to get you, sir.
Hello?
Yes,
Yes,
sir.
sir.
Right now, which one empty?
A site is empty.
Right, A site empty. Just wait.
You didn't listen the question.
Parvin? Any doubt?
No, sir.
Tell me, Parvin.
No doubt.
Right? So this is the final step in elongation. Is it clear? So first of all, actually, this is methionine tRNA. This is methionine tRNA. Initially, this methionine tRNA placed at the peptide site, right? Hello?
Yes, sir.
Second tRNA placed at A site, right?
Yes,
Yes,
sir.
sir.
Initially, this E site empty. But
Yes.
after that, this translocate this first tRNA to E site. E site simultaneously when the first methionine tRNA transfer to the E site, automatically the second tRNA where the existed in A site, this is also translocate or shifted toward the
So P site.
peptide site. That means P site. P site. Now this E site with help of first tRNA, P site with first amino acid with second tRNA. Now the A site empty. Now
Yes,
what
sir.
would be? What would the next reaction? The same continuation, like the second
Next amino acid tRNA
right,
will come
next
and bind at
amino
A site.
acid, next amino acid 3, for example 3 with the tRNA,
Leucine example.
right? Next same E site, and this is shifted to the P site, it would be empty. Is it clear?
Yes, sir.
And this process continued until reach the termination factors. Is it clear first?
UAA, UAG. Yes.
Right. UAA, UAG, UGA.
UGA.
They are the termination factor. I will tell you later. Before termination factor, let's see how the bond formation between first amino acid to the second amino acid. Right?
Okay.
Right. Formation. Hello.
Your
Hello.
voice is not audible.
Now clear?
Yes, sir.
This picture clear?
Yes, sir.
Yes, sir.
Right. For example, this is first amino acid, methionine. Methionine. This is second amino acid. Here the peptide bond formation between first amino acid carboxyl group is COOH carboxyl group, and second amino acid amino group. The amino group. So carboxyl OH, amino H in the form of water leave. Remove water and form as a peptide bond.
K2.
Between, right. First and second amino acid. This is peptide bond. So like this for first and second. And if you add the third one, it would be continuous same. That means already here the carboxyl group. Already here the carboxyl group. Next third amino acids, the bond
Amino
formation
group.
between carboxyl group. Next that's it. Amino group. Like for next add another 4 amino acid, 5 amino acids, 6 amino acids, so many. So for each and every amino acid, for first to second carboxylic and here the group formation, peptide bond formation. Peptide bond formation like this. This type of this peptide bond called as polypeptide because more than 2. That's why it is called polypeptide. Is it clear? Hello?
Yes, sir.
Is it clear?
Yes, sir.
Right. Now on termination. Termination, first of all, remember when the ribosome reach the process would terminate.
Voice is not audible.
Right. When the ribosome units reach the nonsense codons, the process would be terminated with help of RF1, RF3, and GTP. RF factor, RF3 factor, and GTP energy. With help of GTP energy, RF1, RF3 factor, with help of nonsense codons, this translation process would be end. Is it clear? Hello?
Yes, sir.
Right, just wait. Please join again after 3 o'clock in second session, okay?
Okay, sir.
Before that, just let me complete the end one. So this is 1 peptide bond formation, 2 peptide bond formation, 3, 4, plus 10 amino acid group form. Now this reach chain is the UAE nonsense codon. Now automatically RF1, RF3, GTP activated, bind this region, this black area, completely blocking this area like this. Now this peptide cannot move forward. Cannot move forward as the blocking with help of RF1, RF3, GTP. Now it would be this peptide chain would be removed, right? Hello? Hello?
Yes, sir.
Sir.
Is it clear? Is it clear?
Yes, sir.
Right. No, and join again in second session. Plant molecular biology and biotechnology paper. Okay.
Oh, yes, sir.